So we have in this presentation with us Dr. Stuart Rees. And Stuart, I just need to ask you a couple of questions of bio. And you have doctor in front of your name. That is a doctor of what?
That's a medical doctor, Gary.
Okay. And what do you do as a medical doctor?
Well, I'm a GP or family physician, so I see ordinary general medical patients, and we have a large practice in addiction medicine as well.
Okay. And you appear within your publications as having professorships at various unis. Can you just fill us in a little bit on that front?
Yeah, that's the University of West Australia and Edith Cowan University. They're both near Perth in West Australia.
Okay. And in this video, we're looking at cannabis population studies. What was it that led you to population studies in the first place, to put your emphasis there?
Well, as I said, we've got a lot of drug-dependent patients who take all sorts of drugs, but for virtually all of them, their drug-taking career started with cannabis. Which made me worried about what the effects of cannabis were as a gateway drug. The other problem is that I've got numbers of families. I've got a huge number of autistic children in my practice, and there's a lot of women have got all of their children, 3 out of 3, 4 out of 4, 5 out of 5. They're all autistic, and all these women share in common cannabis use during their childbearing years. So that made me worry, and the question became, is this just idiosyncratic? Is it just unique to my practice, or is this what the whole is happening in the wider population? And so to do that, we needed to look at large population studies on whole continents. So that's why we went to the US, where this stuff is best documented.
Okay. And back in 2016, you and Gary Hulse, another academic in this case, got a fair bit of publicity. And I saw something in the Daily Mail about some breakthrough. What was that?
Yeah, well, we found that autism trends and cannabis trends were closely linked in the US, and we were able to show that they were statistically linked. But perhaps more importantly, there were mechanisms. There are multiple mechanisms by which cannabis disrupts brain development, which might lead to an autistic pattern in growing children, and that's what worried us. And then we published 3 or 4 times on this subject, and we looked at space-time relationships. So with taking space and time into consideration, we found that cannabis and the autism co-vary across space and time together. And we're also able to show statistically that that was causal, as well as multiple causal mechanisms, as you'll hear in a few minutes.
Okay. And how many peer-reviewed studies would you have had published over the years that actually deal with the kind of mechanisms you're going to talk about tonight, the broad mechanisms, and your use of statistics, your methodology there as well?
Oh, well, there's several questions, Gary. We've got 4 on the subject of autism. We've got another 4 or 5 on the subject of disordered brain development and mental health. We've got our textbook coming out in the very near future, which deals with all this in great detail. And there's 160 papers all up. We've got about, I think it's 80 papers now on cannabis, with another couple just going into the journals. They'll be out in the next few weeks, I imagine. So it's going to be about 84 in a month or two, or just on cannabis, and a lot of them using these kind of advanced statistics we'll mention today.
Okay, and what is the caliber of the journals that you have put those studies into?
Well, some of our work's been published in Science just a few months ago, some in New England Journal, the top medical journal in the world. We've had several published in Addiction Biology, which is one of the top, I think it's the top drug addiction journal in the world. We've had papers published in the European Archives of Psychiatry and Clinical Neuroscience, which is one of the top neurology journals in the world, neuroscience journals. And we've had, lots. There's several published in the Journal of Xenobiotics, which is a high-ranking journal, impact factors 7 or 8 now. And there's quite a lot in the MDPI group. There's lots and lots. All the European teratology stuff went mostly to MDPI, I think. So as I say, we've got 70, about 85 papers will be soon. And that bibliography is available from me. I can flick it to you if you want to make it available to the viewers of this video.
Okay, so that adds up to a lot of peer-reviewed studies. A lot of people have looked at your work, have looked at your methodology, and so it is very significant. And I think there's fairly good trust in the conclusions that you will come to. So over to you, Stuart. Talking about autism, this is the first of a number of presentations that we'll be doing, and we'll be releasing these through our Drug Free Australia channel and our One Voice, One message channel as well. That'll be on YouTube and Rumble. Over to you, Stuart.
Thanks, Gary. Well, today I'd like to talk about autism, centered mainly in the USA. And we're going to talk about the epidemic, what that looks like, and also some of the mechanisms which account for what we actually see in the field. So let's start talking about the epidemic. And this is the US autism figures. As you can see, it's growing exponentially, and we've got the references available down there. Now, this is a map graph of the rates of autism across the US. You can see yellow, white is low and black, purple is high. And you can see that Minnesota, Maine, Oregon here are high. And you can see the development of this pattern gradually across the US in that way over
Okay,
time.
can I just point out that 1992, that was the year that cannabis use was at its lowest. It had been higher, and it came down because of the Just Say No program of Nancy Reagan, president's wife. And then from that point, it went up. And so 1992 is a very significant starting point, is the top map that you see on your top left screen. And then that works its way all the way down to 2011. So just pointing that out, Stuart.
Yeah, that's right. Now, I should say that the autism takes 8 years to diagnose by definition. They don't diagnose until the children are 8 years old, which means that you're always well behind the data plan. Then it takes a couple of years for the data to come out and get analysed. So, here we are in 2025. This paper is a couple of years old, this work, but that's the reason for the big temporal lag.
Yeah, and in 1992, there just wasn't a lot of autism. So there we go.
Yeah,
Yeah.
and then President Trump mentioned that a few days ago. You see the news clip? He said it's gone from whatever it was, 2 in 10,000 to 1 in 31 now. And the whole US cabinet just came out a couple of days ago, is very distressed about this. They don't understand what's going on. And this is exactly what's going on this slide. This is a bivariate autism map. You see here we've got cannabis here and autism here, and where it's pink and purple both co-vary up and they're both high together, and green is where they're both low. And you can see here, this is Massachusetts, that's Michigan, that's Oregon, California. That with time, as the cannabis goes up, the autism co-varies and go up. That's I think that's the state there, which might be Vermont or New Hampshire. I think it's Vermont there. So you can see this pattern developing where they're both co-varying together.
Can
And
I
you can,
just go back to the one, Stuart? It's, for people who are not used to these kind of things, this is a synthesis, if I'm right, and you just correct me, this is a synthesis of two lots of statistics. This is rising cannabis use and rising autism rates, which we saw on the previous page. And so just to make it clear for our audience, which is the colour that shows the worst results for any state? So you've got a little colour thing there on your screen, mauve.
yeah, the pink and purple up here, the hot pink ones.
And
So
if it's
see,
green, that's the least
yeah,
of
the green's the
the
list.
correlations. Okay.
Yeah. So where these pink and purple ones is where they're both high, and that's what that shows. You can see the emergence of these pink and purple colors with time.
Yep.
Actually, that's Vermont, and that's New Hampshire beside it, and there's
Yep.
Massachusetts. There is pretty pink.
Okay.
Now, what you can do is you can look at the rates of the US states by cannabis legal status. So you've got the illegal down the bottom here, the medical here, and then the decriminalized at the top. And this shows a clear trend from the illegal to the decriminalized are higher, and that's up to 2011, which is the data. And you can use statistics to show that with time, as those trends are projected out, there's 2025. And by the time we get to 2030, it's 60% higher in 2030 in the decriminalized states than in the illegal states. So that's a massive jump. And that correlates directly with cannabis legal status, which of course determines cannabis availability and the penetrance of cannabis and cannabinoids into the population. So
Hmm.
the 455 over 287 is a 60% higher jump, and that's a mathematical projection of where it's at today. Now, this is the change 1995 to 2011 by cannabis legal status. This is illegal, medical, and decrim. And you can see illegal versus decrim. There's a significant p-value. In science, p less than 0.05 is significant, and that's 0.01 is a lot less than 0.05. This is called a box plot because they look a bit like botches, like boxes. And these notches, you see the notches, that gives the confidence interval. And where
Yep.
the notches don't overlap, that shows statistical difference. And you can see that notch and that notch don't overlap. And that's why the pink one and the blue one are statistically different. That's what that's saying. And then if we dichotomize by illegal and liberal, again, these notches don't overlap. P is significant. And then we dichotomize by changed and unchanged, which is changed. Unchanged is obviously illegal, the historical status, and changed, and again, P 0.046. So you can see that the cannabis legal status directly affects the frequency of autism in the US.
That's very important. Okay.
Now, this is a spatial regression study. This is raw data. It's a bit of a busy graph. This is the reference to our paper, European Archives of Psychiatry and Clinical Neuroscience, that we mentioned before. And you can see that this is spatial regression taking space and time into account. And when you take space and time into account, you've got cannabigerol here, cannabigerol there, THC there, THC, with highly significant, see P less than 0.05 in all these different models, 2 lags, that's 2 years lag, 4 years lag, and different instrumental variables. And you can see for all these ones, see P less than 0.05 is significant. Here's P 10 to the minus 10, 10 to the minus 9, 10 to the minus 6, 10 to the minus 5. So you've got a lot of these. This is the ethnic cannabis exposure. You've got a lot of these parameters, as we've indicated, are very highly significant. This is on a space-time regression
So
system.
just give us a helicopter summary of what you're reading out of these figures here. What are they telling the audience?
That's showing that the autism is varying across space and time with the cannabis, and the cannabis is tightly related to the different measures of cannabis exposure to the autism incidence.
And this is broken out by racial groups as well.
Yeah, so these are all the drugs and these are all the ethnicities. When you put everything together, you've got drugs, cannabinoids, ethnicities, income, all the races. The cannabis is persistently significant in the
Okay.
final models.
And so people will ask, how can you break this out for specific cannabinoids? Like when we talk cannabinoids, these are constituent cannabinoids within cannabis that are all distinct from each other, that each have different effects. But how can you actually work out what cannabinoid is doing what?
Well, the US publish from University of Mississippi, they publish the cannabinoid concentrations of the Drug Enforcement Agency, the DEA cannabis seizures. And that gives you the average concentration of cannabinoids across the US. So we don't have measures of cannabinoid exposure by state, but we can estimate it from the cannabis use in the state multiplied by the cannabis concentration that's available nationally to give us an estimate of the cannabinoid exposure by cannabinoid in each state. So these are
Yeah,
calculated estimates, and it should be pointed out that averages can cover a lot of wiggle, and obviously that's an area for further refinement, but getting significant samples from each state is a big ask. So this is just a computational
it's a guide.
estimate in for each state.
Yeah, good. Thanks.
Now from that, you can calculate what we call e-values or expected values. Now, expected values is a rate of the confounding. In other words, could this association, which we're describing here, between autism and cannabis, could it be due to something else, like vaccines or something like that? And there's a mathematical way to adjust for, we call them extraneous confounders, for other stuff that could be replicating what looks like a causal relationship.
Mhm.
And the answer is for these expected values or e-values, if it's more than 1.25, the convention is that's regarded as being causal. So anything more than 1.25, the relationship between tobacco and lung cancer is an e-value of 9, right? Which is a lot
Oh,
more than 1.25. And 9 is regarded as high, right? So anything above 9 is high or very high. So that's
all
the
right.
scale.
So what you're seeing in your tables here, you got cells with a whole lot of figures. How high are these figures?
Well, some of these figures go up to infinity. That's 10 to the 27. And here's 1.3, 1.16, 1.71, 10 to the 25, 10 to the 29, infinity. And then the e-values have a lower bound, they're like a 95% confidence interval. And you can see they're given here, and some of those are very high, 10 to the 9, 10 to the 5, 10 to the 4, 5.
So you're talking orders of magnitude above 1.25.
Yeah, you're talking
It's not just 1.25 and a bit higher. This is orders of magnitude higher.
yeah, right, all the
10
way up.
times,
Up
100 times.
some of them are on infinity. That's right. So you're
Yeah.
talking this the and you talk to the people and this comes from the top epidemiological schools in the world in New York, and they say these very high values they're only seen in causal relationships. Think lung cancer and tobacco.
Okay. And this is the real important thing about these things which are called e-values. It's all about causality. So we know that there are correlations that you've been able to show, but now you're trying to get down to is there a causal correlation? And people say, oh, well, correlation is not causality. But then wherever there is causality, there is correlation. And
Yeah,
that's really important.
yeah, but that argument about correlation doesn't equal causation. This slide here is not just a multiple regression. This is a space-time regression,
Yeah, so
right?
it adds
So
a
you're
whole lot.
saying not only are they related, but
Yeah.
they're related in space and time. In other words, as the cannabis goes up, the autism goes up there. So it's closely related across space and time, which is exactly what you'd expect to see in a causal relationship. And
Okay.
then, so we've got hugely significant p-values, and the e-values are just going right off the top end.
So
We
it's
can say,
very significant.
yeah, highly significant. So this is not just association. They say that the rooster crows in the morning and the sun comes up. That doesn't mean that the sun doesn't come up because the rooster crows. The rooster crows because the sun comes up. But that's reverse causality. But this is nothing like that. We're way beyond that with these very advanced techniques.
Yeah, cool.
So let's talk about the patterns of European brain birth defects, because what this is implying is that cannabis is interfering with normal brain development. So the question is, how much does cannabis interfere with normal brain development? And this is called meningomyelocele, which is one of the significant birth defects. There's a hole in the back, and this child was aborted, as many of them are, for that. Now, this brings us to, this is looking at Europe. Now, what this is this has got 6 birth defects along here. You can see anencephalus, that's no brain, encephalocele, nervous system, neural tube defects, severe microcephaly, and spina bifida. So there's 6 birth defects here. And here we have tobacco, alcohol, a cannabis index, amphetamines, and cocaine. And remember what I said, if you've got lung cancer on the y-axis and tobacco on the x-axis, then you expect to see a straight line pointing upwards to the right because they're positively related, right? So you're looking for those straight lines pointed upwards to the right. And this is a kind of paneled grid plot. And what you see is that for most of these defects with tobacco and alcohol, it's mostly pretty flat with 1 or 2 exceptions. And for amphetamine and cocaine, mostly pretty flat. Maybe there's an exception there. But for cannabis, most of the lines are pointed upwards to the right for 5 of these 6 defects. So you can see that cannabis is the standout teratogen just based on this simple scatter plots. And then we take another 5 birth defects. This is no eyes or tiny eyes. No, yeah, this is no eyes at all. This is craniosynostosis, which is a fusion of the skull bones. That's eye abnormalities generally, and that's hydrocephalus. And this is the same thing, right? So you've got tobacco, alcohol, cannabis index, amphetamines, and cocaine. And for some of these defects, again, the relationship with cannabis is quite strong. For a couple of these, it is with the other drugs, but in general, it's unimpressive compared to the cannabis, which just on these 2 plots shows that cannabis is the heritage you watch, and the others is sort of really a sideshow.
So when we're talking about these particular birth defects, how many of these, like, you've got eye issues, you've got small brains, you've got no brain or virtually no brain, and, on the previous slide and so on. How many of these are likely to self-abort, just to miscarriage?
Well, a lot of them are like anencephalus. A lot of those die prior to birth. That's no brain. But a lot of them die within an hour or two. Most anencephalics die within the first day. A lot of these are aborted. It's called ETOPFA, which stands for early termination of pregnancy for
Yeah.
anomaly. So the doctors, will talk to the patient, and most of the women will choose to abort early because the kid's not going to make it or it's too severely damaged, as we saw in the slide a moment ago. And now that's important because we don't have good measures of that ETOPFA by anomaly practice at the national level. In other words, most of these defects are going to fly under the radar because we don't have measures on the total. We only have measures on the live births, not the whole of the thing. And as you say, a lot are aborted spontaneously, and a lot of subject to ETOPFA. So that what we see is could only be the tip of the iceberg for many of these severe defects. That's very important to keep in mind.
Very important. Keep going.
So here we're looking at all these are various different indices of cannabis. Europe has more indices of cannabis than America. So we've got last month cannabis, cannabis herb, cannabis resin, daily cannabis interpolated, last month cannabis times the herb concentration, last month cannabis times the THC concentration of the resin. And this is the last month cannabis times the herb times the daily use interpolated, last month cannabis times cannabis resin,
So
daily.
can I slow you down on that one? The first three are
Hmm.
different forms in which cannabis comes, right? You've got,
Cannabis herb, and it's what they
yep, so you
say.
got herbs. Sorry,
Cannabis
the second and the
resin.
third.
Yeah, the first one is last month. How many of them used cannabis in the last month? So these are
Yes,
different
sorry, that's right. And second and third are different,
forms.
forms in which cannabis comes, resin or herb.
Yeah,
And then you get to use again.
daily cannabis use, and then you get the products. So these are compound metrics derived
Yeah.
from products of these other ones.
So
So
they're combining these other ones that are
yeah,
on the left.
yeah. So,
Okay.
so that's along the x-axis down the bottom, and then you've got the rate of the defects up here, and you can see that particularly on the right, some of these compound indices are capturing the relationship with the birth defects quite well. There's quite a strong signal, and it's often built by multiplying these different cannabis metrics to get these strong signals that you see here on the
Again,
right.
it's a line that's going up towards the right. And what we've got is six birth defects down the right-hand side,
Yeah.
which were the six that were on a couple of slides back.
Yeah, and we do this
Okay.
the same for the other five, and you can see that four of the five have this strongly positive relationship here. Last month, cannabis times herb times daily use interpolated, and you get these strongly positive signals with these compound indices.
Like more than strong. But anyway,
Yeah, they're very strong. Now, this is a map graph, right? Okay, this is like a heat map of Europe, 2010 to 2019. We saw earlier for the US, and this is Europe. This is the decade we have the data for. And you can see this is the central nervous system anomaly rates over time. You see France is a pretty hot spot. Bulgaria is black. It's high. Um, and that's the main standouts there. That's all the CNS. This is severe microcephaly. Now, remember, everyone was very upset about microcephaly from Zika virus. But when you look at it here, you can see it's high in Germany and France and those ones. Germany's black there. Bulgaria's black. Spain there, pretty bright. So you can follow that over time. This is anencephaly. Now, remember, there's two papers from the CDC, Atlanta, Georgia, linking cannabis with anencephaly. And they say it's a causal relationship. They say
just,
it's about double.
just remind me what anencephaly is that. Is that a tiny brain or no brain or
No, that's no brain.
no
No brain.
brain?
No.
Okay.
This is babies born without a brain. And you can see that Bulgaria is high there. France is pretty high. Netherlands is high, and that's in that one. So that's what that shows. And then this is neural tube defects. That's spina bifida and encephalocele. Again, Bulgaria high, France pretty high, Germany high in some of those shots.
Okay, with neural tube defects, what's the effect going to be on the baby if it's born?
Well, some of them it's mild and they can be repaired, and some of them it's severe and they can't be repaired and they can die. But
Okay, so there's going to be grieving parents
yeah,
or parents who've got a bit of money to find, for a few operations, right?
right, yeah, and some of them don't
Okay,
get that. Now
yep.
this is the bivariate plot again. This is
This is
that triple
putting two statistics together, use
triple index we
and
talked about
disease
before.
burden. Okay.
So this is all the central nervous system anomalies, and you can see that there's the pink, remember, and the purple is both high. You see Bulgaria, Netherlands, France, they're both high, and you can see the emergence of that. Spain goes up, but then it goes down. So you can see the emergence of that across time. And this is severe microcephaly. So this is severely small head, which obviously means severely small brain. You can see France, there's Spain, and Netherlands, and some of those shots stand out together, and that's neural tube defects. You see France is coming up there, Netherlands and Bulgaria is showing there. Now, when we look at the nervous system in the geospatial analysis, again, on all these different models, the cannabis is highly positive. Remember, 0.05 was significant, and this is much less than 0.05. And this is a space-time model showing that across space and time, the defects are changing with cannabis.
So
Neural tube
you were looking at tobacco, alcohol, that kind of thing against cannabis in that last one.
yeah,
Yep.
this is
Okay.
tobacco, alcohol, and everything, and income, I
Yeah.
think, here as well. But the income's in some of them. But the point is that the cannabis persists in all these final models. When you dump all the stuff that doesn't matter, in most of these models, you're left with one of the metrics of cannabis. And this is the E value pattern. And, we saw it before. Look at these massive E values, 10 to the 33, 10 to the 28, 160. This is 40,000 there, 4,000 here, 100,000 here, 10 to the 16 here. So the E values are massive, and the 95% lower bound is also very high. And this demonstrates causality at these very high levels.
So that's causality, and it's like extremely high. So anything over 1.25 is significant,
Yep.
and you've got massive, massive, massive out of this world figures there.
Yep.
So yeah, keep going with this one.
So this is the E value list from all of those studies. And as you can see, there's 50 E values. 44 are greater than 9 of the estimates, which is 88% are in the very high range. And the minimum 72% of the very high range, more than 1.25, 100% of those E values are more than 1.25, and 96% of the E values are more than 1.25. And
So
that, you're looking at causality there.
so cannabis is really implicated as causal on those tables.
Yeah,
And
yeah. And remember, these are major brain defects. This isn't autism where the kid's normal, but the brain's a bit off. This is major brain defects. You don't even need to be a doctor or a nurse to see there's something. So the point is that cannabis causes these major defects. Obviously, there's going to be a spectrum, and there's going to be mild, moderate, and severe. And the autistic people, we're saying, are up the mild end.
yeah, I'm getting that.
So,
Okay.
so this is looking, this is a study from a very powerful, it's called an epigenome-wide association study, right? Everyone knows what the DNA looks like. It's a spiral helix. And
So you're getting into mechanisms here?
yeah,
Yeah.
it's got little switches on the DNA, which are called methyl groups, which turn the genes on and off. And that's why a heart cell is different to a blood cell is different to a muscle cell, because different genes activate it.
Yep.
And this software that's on the genes are called the genome. The software is called the epigenome. And this is an epigenome-wide association study, which scientists abbreviate to EWAS. So this is an EWAS, epigenome-wide study, looking at the different receptors in the brain. And here you've got the two main stimulation receptors and suppression receptors, glutamate and GABA. And these are the genes which control glutamate. These ones control GABA. And you can see in this EWAS, these are called EWAS hits. They're hits on the genes that control these key receptors that turn the brain on and off. And here's the dopamine receptor and the dopamine pump. These are the two of the opiate receptors here. And these are the two of the key genes that control the scaffold that hold the synapse together. And the point is they're all associated with highly significant p-values on the EWAS hit. In other words, the epigenome is knocking these genes off epigenetically at the software level, these key genes of brain function, and this is after cannabis exposure.
So that's cannabis that is doing that damage to those particular
Yeah.
receptors.
Yeah,
Okay.
this
All
is an
right.
EWAS study in mice, and it's been validated with the results in humans for this
Okay.
fantastic group here from North Carolina. Now, these are the opiate receptors that are mucked up. This is the mu receptor and the delta receptor, and you see P is highly significant here. That's the raw P, and that's adjusted for multiple testing, again, highly significant. This is DSCAM, Down syndrome cell adhesion molecule, which is a key molecule which forms the synapse and makes the synapse stick together and makes the synapse work. You see, there's all these significant P values here in the synapse, these are epigenetic hits.
So
And
you're
this,
saying that cannabis is tied up with Down syndrome on that table?
well, this is the Down syndrome cell adhesion molecule, which was first identified in Downs, but it's now been shown to be a key molecule holding the synapse together, a structural molecule in
Okay. All right.
all synapse, not
I'm
just
with you.
Downs, right?
Okay.
And here's the retinoid receptor, which, everyone knows humans have a big forebrain, a big cerebrum, and the retinoid receptor is key, one of the key things that makes it so big. And this shows cannabis mucking up at the retinoid A and B receptors to at the high levels of statistical significance, both before and after adjustment for multiple testing, to destroy the retinoid function in the brain. And the same thing here with Robo. They call it Robo. The full name is roundabout. It's a gene. This gene is responsible for making the human forebrain grow so big. And this shows that cannabis knocks it off. It interferes with brain and forebrain development, at high levels of statistical significance, as you can see. So let's talk about mental illness, which is another form of disordered brain function.
Okay. What do the red dots mean?
The red dots are the substances that they've been exposed to, and the blue dots are the illnesses. So I can go through those if you like and just show you what's what.
Yeah.
Um, so this is cannabis, cigarettes, alcohol, cocaine, and heroin. And the illnesses are depression, any mental illness, serious mental illness, major depressive disorder, suicidal thoughts, suicidal plans, and suicidal attempts.
So you're going to work out correlations between those substances and those illnesses,
Well,
right?
if you look at it in the 26 to 49-year-old age group, across all these diseases, they're going up. And if you look at cigarettes is coming down in that age group, alcohol's
Yep.
coming down in that age group, cocaine's coming down in that age group, heroin is only 0.5%, so that doesn't count. But they're all going up, and the only one that's going up in that age group is cannabis. So this shows that in that age group, cannabis is rising, all the mental illness is rising, all the other substances are flat or going down. So it must be cannabis just looking at that.
Yeah.
And it's driving all these mental illnesses up by itself, and everything else is falling. It's very powerful montage of this is the director's graphs. This is what the director actually showed in 2017. Now, this is a map graph again. This is 2010-12 here. This is sub-state areas data from SAMHSA, Substance Use and Mental Health Service Administration of the US. So 2010-12, 2012-14, 2014-16 are here. And if you look at it, see cannabis use is emerging. So here you get this northeast, northwest, and Colorado in the middle pattern there, and Alaska. So that's the northeast, north. So you get a geotemporal trimodality pattern.
So
And then you
that
look at
left column is the cannabis use, and then our next three columns are
it.
different kinds of illness that we're
We've got
talking about.
any mental illness here, serious mental illness here, and suicidal thoughts here.
Yeah.
And if you look at the geotemporal trimodality, see there's a bit of a concentration in the northwest, something going on in Appalachia, and in the northeast, and then here in the northwest, there's something going on, and you can see that
Yeah.
mental illness
Oregon.
changes a bit over
Yeah.
time.
Yeah.
With serious mental illness changes a bit, but suicidal thoughts, you can see the northwest, including Alaska, the northeast, uh, and Colorado have gone right up into the black zone, paralleling exactly what the cannabis, this very similar pattern to the cannabis.
Yeah. So you're going to fuse these graphs together, I take it, and synthesis. There we go.
There's the synthesis. So there's any mental illness, serious mental illness, major depressive illness, suicidal thoughts. And you can see the pink and purple emerging, the north of California, parts of Colorado, Alaska. That's suicidal thoughts. And this is the depression. And so you can see these patterns emerging here.
Yeah. So Colorado wasn't looking good.
Now,
And certainly up in the north,
and Alaska's
yeah,
been legal
and
for a while too.
yeah, okay,
So legal status and moderate relationship. Well, obviously, serious mental illness is the one to focus on out of all of those. And as you can see, this is modeling. This is so in the model relationship using statistics and mathematics, you can model the relationship between mean last monthly cannabis use by the deciles of cannabis use, 1 to 10, and the serious mental illness rate. And you can see there's this relationship which rises as the cannabis rises. And then if you look at it by legal status, just as we did for autism before, you see there's a relationship between medical, decriminalised, and legal, and the notches don't overlap. These two here, they look different, but the notches are overlapping, so they don't count. But when you get up to legal, the serious mental illness takes a jump above all the others.
okay.
And this is the spatial matrix we use for space-time regression. We have to do that. And so we come back to space-time regression again. And this is a log of the cannabis, serious mental illness, in all these different models, simple models and more complex models. And the cannabis is persistently positive at high levels of significance. See 10 to the minus 12, less than 10 to the minus 16, 10 to the minus 10. High levels of statistical significance here for the mental illness, serious mental illness, in a space-time consideration.
And that's tied to cannabis using states where it's
Yeah,
legal or
yeah,
decriminalised,
that's right.
whatever. Yeah,
That's right.
okay.
That's right. So here we get the E values, and you can see 790, 4, 5, 19. This is 10 million, and this is 100, 700 million.
So that's causality again,
So that's
and
causality on the E values.
yeah, some of them through the roof.
Yeah, that's right. Now
Yeah,
let's look at mechanisms, because remember we said before the rooster crows in the morning but doesn't make the sun go up. It depends if there's a mechanism, right? The rooster crows because the sun goes up. The mechanisms work in the other way. So the mechanisms are key to the causal argument. Right? If there's a mechanism linking it, then the association can be seen as causal if it's reasonable.
really important, Stuart. That is like, absolutely necessary that you be able to work out the mechanisms.
That's right. So let's talk about brain development. We're going to do this under 3 headings. We're going to talk about morphogens, what the scientists call morphogens. We're going to talk about synapses. We're going to talk about the epigenome. Now, we'll talk about the morphogens first. Now, the morphogens, imagine the little baby inside the mother is being woven together by a gradient of chemicals that's guiding and directing all the cells where to go and what to do. So it's like a big loom inside the mother that's in 3 dimensions, and it changes across time. So it's a 4-dimensional loom that changes, but it's got the woof and the warp just like a loom has, a weaver's loom has. And the point is that cannabis disrupts nearly all of those morphogens. In other words, if you take the woof and the warp and bend it all out of shape, then you cannot have a normally developing baby because you've just corrupted the loom that weaves the baby together. You understand that? And that's particularly important to brain development. And in this illustration, we can see that the anterior part of the developing baby, and this is the head and the brain, and there's all these different morphogens here. And cannabis knocks off that one, that one, that one, that one, that one, and that one, and that one. So 7 of the 8 there. Um, and these morphogens control the folding of the embryo and the way the brain develops and the forebrain develops. It's all controlled by morphogens, and this is the rhomboid segments at the back, which are also controlled by morphogens. And by messing up this loom, it messes up brain development. Now, this is a picture of the spinal cord, and you can see the main morphogens acting on the spinal cord: bone morphogenetic protein in the wince, and at the front, sonic hedgehog. And cannabis messes up that, and that. Now, these morphogens control exactly which cell develops where and how it activates and what it turns into. This is very delicately controlled across time and across space, these morphogens. And
So
this,
that explains your neural tube. What you
yeah,
had
yeah,
defect,
yeah,
okay?
yeah,
It
yeah,
slides back,
yeah,
and this explains why that happened.
yeah,
And I take it it's early in the life of a fetus that this all happens.
that's right. Now, then we come to this big human forebrain we're looking at here, which is here. Now, it turns out that the forebrain in the lower animals, the neural stem cells divide down here that make the brain cells. They divide and then they become a nerve cell, and that's how they get their brain. But in humans, that doesn't happen. what happens is the nerve cells divide, they're called radial glial cells, and then they divide and divide and divide and divide and divide. So the brain cells become several generations later, and you get this, huge exponential growth. That's why the human brain is big. So anything that interferes with that divide, divide, divide process, it's called indirect neurogenesis. Anything that interferes with that will limit the size of the brain development.
Okay.
Okay, so that's what this is a picture of. And you can
Got
see, you can see that the cells are guided by the radial glial cells, and that's how you make a brain. And the brain has a very delicate structure. This is a picture of the hippocampus, which is folded, that where the memories form. And you can see this is under here is where the cells, the baby brain cells are born, and then they migrate across the brain. You can see it illustrated in different ways in these shots. And this is what happens. The brain cells come out of deep inside the brain and they migrate across the top into the cortex. So they go on this big migratory path. And this, of course, is controlled by the morphogens. It's controlled by the genes and the software that runs the genes, which is called the epigenome. And so by mucking up the morphogen lattice, by mucking up the genes in the epigenome, this is why the brain doesn't develop properly. It interferes with neural migration across the brain. And here we looked at that before. Oh, we
And
got
it's cannabis that's responsible for messing up those processes.
Yeah.
Yeah.
And this is exactly, remember we talked about direct neurogenesis like snakes and birds have, and indirect neurogenesis like humans have, that divide and divide and divide. And as you can see it down here, low ROBO and high DLL. This is in cells. This is a nature communications in cell. This is ROBO, which we talked about before. We showed how cannabis mucks up ROBO epigenetically. And this is one of the things interfering with human forebrain development, which of course clinically is anencephalus and severe microcephalus, which is exactly what we see when we look epidemiologically.
In the population studies. Okay.
Yeah,
All
that's
right.
right. Now, so here we go, references to morphogen disruption by cannabis. So these are some of the morphogens that we could consider. This is just taken from my bibliographic program. This is not exhaustive because when I'm writing a paper for the journals, they don't want me to put 200 references just on one point. It doesn't work that way. But just looking at this list of morphogens here, you see there's 168 evidence where cannabis interacts negatively with these morphogens. So it completely trashes the morphogen lattice. Do you understand?
All right. And you get all of this data from other people's studies, right? So when it comes to mechanism, you're the population study expert.
Right.
But then when it comes to mechanism, you're going to bucket loads of other studies
Yeah.
to get this information.
This is taken from PubMed, right? The National Library of Medicine in the US Congress. So this is the top medical library in the world. This is all, peer-reviewed medical literature.
Yeah.
Now let's talk about the synapse, the key molecular transsynaptic structures of autism, because autism is often called a synaptopathy, right? The cells aren't talking to each other properly. That's what the problem with autism is. A lot of people think that. See, so here is autism as a synaptopathy, the University of Oregon. There's a problem in the nerve connections between each other at the synapse. And even Wikipedia knows that neuroligand dysfunction is implicated in autistic spectrum disorders, and this is one of the key molecules that gives the synapse structure, right? So Wikipedia know it, but we don't. Now, neurexin has a binding partner, neuroligand, which, as you can see, and here it is, mutations and lesions in neurexin are recently implicated in autism spectrum disorder, schizophrenia, and mental retardation.
Mm-hmm.
So, RFK Jr. and Trump, people who are looking to understand autism, they need to understand the synapse and how it works. And you can see neurexin and neuroligand here together give the structure synapse and hold it together and give the synapse structure and make the thing work so that then the neurotransmitters and the receptors and all the complicated machinery can go to work, but the synapse has an intrinsic structure which is confirmed by neurexin and neuroligand, you see. And you can see here they touch across the synapse in that way. And you see this is more complex. This is a complex of neurexins, a complex of neuroligands. This is the AMPA receptor, the glutamate. This is the workhorse of the CNS, that excitatory receptor, which actually does the thinking and receives the signal from the presynaptic signal. But the synapse itself is held together by the neurexin neuroligand ligand pair. And you say the same thing, transsynaptic complexes of the synapse here, neurexin neuroligand. You can see it all happening. And this is interesting because this shows in some detail. This is called the presynaptic side where the boutons are, and this is released with each neural impulse. and then it's taken up. There's neurexin neuroligin in the synapse. You can see holding it all together. There's a GABA receptor, inhibitory receptor. There's a glutamate receptor there, a stimulatory receptor. But inside the bouton on the postsynaptic side of these key molecules, the postsynaptic density and shank, which gives structure to the machinery of the postsynaptic side. Just as there's machinery on the presynaptic side, there's machinery on the postsynaptic side, and that's held together by structural proteins, PSD95 and shank. And
So
it says
we're taught in high school about transmitter and receiver, when it comes to synapses. So which is which?
these are the transmitters and these are the
Yep.
receptors.
Okay, just to make sure our audience is clear on that.
Yeah, but the point is, it's more complex than that. There's a structure in the
It's
synapse.
the structure is what you're talking about, and
Yeah,
it messes with the structure.
it messes with the structure that holds the synapse together, and it messes with the structure in the postsynaptic bouton. And that's why it says mutations in a number of these genes are associated. SHANK3 scaffolding protein have been implicated in autism. And here we go: neurexin, severe onset epilepsy, profound developmental delay. It's exactly what you'd expect because the synapses aren't working properly. This is the synaptopathy of autism.
Yeah,
And here's the molecular structure of the synapse in more detail. See, neurexin, neuroligin, we've talked about. This is the AMPA and NMDA. These are the stimulatory receptors. This is another glutamate receptor. And here you see shank and HOMA, and there's the PSD, these are some of the key structural molecules of the postsynaptic side, the bouton on the other side. So you would expect problems with the PSD, shank, and HOMA to be expressed in deficient synaptic function. That's what you'd expect, right? Because it's a
yeah,
structure.
yep.
And then if you look at shank and autism, this is PubMed. This is a search just done March 29th. 165 hits on PubMed for shank and autism.
All right. So there is plenty of evidence
Yeah,
out there, plenty of science on this
yeah.
that you're not just making that up.
No, and shank and autism and HOMA, 59 results. And then when you go to the EWAS studies, you find there's lots of hits with cannabis, HOMA, and shank. You see, so you can say, and here's altered cerebellar connectivity to the amygdala and anterior-temporal pole in autism. So there's a problem with connections, which is synapses, right, between the cerebellum and the cerebrum and the anterior-temporal pole. So there's a
So there's a
problem.
whole convergence of various scientific areas of study that all are, you know,
Yeah,
sort of
it's
building on each other.
brain structure and function. And then
Yeah.
this is THC disrupts the microtubule uh, guidance system because the nerves have to find their way to their synapses, right? It's like a homing missile. And the system that controls it is controlled by Stathman, and cannabis messes up Stathman. So it's like think of a missile, right? And it's like having a jamming circuit in the missile. The missile's being guided by the GPS or the radar. You've got a jamming circuit, and the missile's going to miss the target
All
because
right.
you're jamming the guidance system that controls axon head growth. And that's what this very powerful paper is. And this is widely often quoted by Dr. Nora Volkoff, who's the director of the National Institute of Drug Abuse, NIDA at NIH. It's a powerful paper. She often refers to this.
Yeah, I just need to remind audience that THC and CBD are the ones that we know most because they're sort of in the media, and you might see CBD at your local supermarket, and say, oh, that's cannabis. But some people get mixed up and they go, oh, what's THC? And that's the psychoactive element within cannabis. CBD is the non-psychoactive element, and then you've got a whole range of other cannabinoids in there. But anyway, keep going.
Well, that's important, Gary, because all the cannabinoids have been implicated in the genotoxicity. And we'll talk about that when we talk about
That's important.
genotox, which is another talk. That's very important.
Yeah,
Now,
yeah.
this is a study from Sydney looking at connectivity in the brain, basically showing an 84% reduction in this circuit, memory to fimbria, and an 88% reduction in memory to splenium. In other words, showing that the brain gets unhooked. So it interrupts brain communication within the brain in a big way, which is part of what you expect with the synaptopathy. The cells aren't talking to each other properly.
So that
But,
explains stuff that we've known for decades.
yeah,
Is that cannabis is messing up with cognitive, with children who are born, from cannabis using parents, cognitive issues, all of those kind of things.
yeah, that's right. So, and also we haven't talked about it here, but cannabis damages the white cells that produce this wiring, right? The gray matter, the white matter in the brain is wiring. It's a lot of insulation. You know, when you get electrical wire and there's a red plastic cover on the outside, well, that's red plastic cover in the brain is formed by oligodendrocytes, which come from oligodendroglial precursor cells, which are damaged by cannabis. So you're damaging the wiring. And here, when you do the scans, you see the wiring is coming unstuck. So you can see how the mechanisms are all hanging together. So
Mm.
let's talk about the epigenome, the software. Like I said, the DNA has these little marks on it that turn the genes on and off, which is the software of the genes known as the epigenome. And these switch the genes on and off, as you can see. And those methylation marks on the DNA look like this, and they come off with time. And when we looked at cannabinoids and epigenetics, we got 209 hits. That was a search in PubMed in the end of January.
No shortage of science on this area.
No. Now, when you look at the Safari database, this is the leading database in the world for the genetics of autism. And you find that there's 1,200 genes that are linked with autism. So this is the world's leading database on, it's all annotated on that. And when you look at cannabis and autism, this is at the epidemiological level. The first three papers here, Baranga, Lepow, and Paul, that's 2021, 2022, 2024. This is the NIDA ABCD study, right? This is the NIH study showing that cannabis is linked with autism and similar disruptions, ADHD-like problems in pediatrics. And this was all MRI controlled. So this is fMRI controlled. This is the top technology in the world confirming what we found in Australia. I think we were amongst the first to find it.
That's really important, Stuart.
It's
Because
three.
your autism studies actually went back a bit before that.
That's right.
Yep.
That's right. And then there's all these others, another 13. These are epidemiological studies linking cannabis with autism. Then cannabis and autism epigenetics showing the overlap. There's 15 studies showing the overlap there. And then cannabis and autistic synapses, there's 14 studies showing it there. So it's not just one or two studies. And the point is there's such concordance between the epidemiology, the patterns of cannabis and autism, the epigenetics, the software disruption, the synapse disruption, and obviously the morphogens. So there's a tremendous concordance of all this data pointing the right way. Then we take human-induced neural stem cells and we get acute and chronic exposure. And they find significant disruption of the synapse, the mitochondria, and the glutamate signaling. This is with induced human neuropotential. So these are neuroblasts, these are nerve stem cells.
And this is THC. Yep.
Yep. And the genes associated with autism and intellectual disability, which is exactly what Trump and RFK Jr. are concerned with. And it's induced by THC, showing in a lab petri dish with everything controlled that it's THC that does this. And this is a fascinating, it's a very complicated slide, and I apologize for that. But this is looking at the acute effects of THC administered short-term, long-term, and look at all the overlap. And then this is COX-9. This is the grid 2 here. So this is the stimulatory glutamate receptors here. And then this is the electron transport complex. This is how cells make energy. Right, cells make their energy in a mitochondria.
Mitochondria. Okay.
And the mitochondria has 5 electron complexes. And as you can see here, 3 of them are disrupted there by acute cannabis, and 4 of them are disrupted there by chronic cannabis. That's what's
So helicopter view explanation of what you're seeing.
Yeah, it disrupts the receptors, like we said before, and it disrupts the energy generation. And everything that nerves do takes energy. It takes energy to send pulses. It takes energy for the genome to work. It takes energy for the receivers. And if you muck up the energy, the whole process falls over and the cell can die, because neurons are operating at about 95% of their max, metabolic rate, using energy to busily do what they do. And if you interfere with that, basically it's good night Australia for the neurons.
Okay.
And this is looking at the postsynaptic density. Remember we talked about HOMA.
Yep, it's your structures now.
HOMA and Shank.
Yep.
Shank, there's HOMA. These are the fold changes. See, -4 for Shank, and we talked about it as a synaptopathy. And HOMA is -2 times fold change, chronic THC. The p-values are highly significant. And these are all the ion channels which are disrupted, because you've got ion channels which conduct the signal at the synapse, but ion channels which keep the nerve cell depolarized, potassium and sodium along the axon. And you can see there's a lot of fold change here. There's big depressions here, 7-fold here with this potassium channel. And this is looking at gene clusters. This is really expressive of HOMA. So you see HOMA untreated. This is the control. Acute and chronic years. This is HOMA that was the key structural protein in the postsynaptic buton. You can see it's dropped off with the chronic PHC. And then the chromatin, this is chromosome organization. This is the mitochondria, cell proliferation, cell signaling here, mitochondria, the RNA, that DNA forms RNA forms protein. And this is development. This is the cells that control the nerve stem cell growth. You see, these are clusters. This is looking at clusters of genes that are messed up by THC.
So the THC from cannabis is throwing a wrench in the machinery, basically, isn't it?
Totally, totally. And
Yeah.
this is the gene overlap. And the scientists know that there's a lot of overlap between autistic genes, schizophrenic genes, and intellectual disability genes. And this is THC and autism. And you can see at the genetic level how there's so much overlap between these syndromes. This explains why cannabis is linked with schizophrenia, intellectual disability, and autism.
That
And
explains a lot,
and
particularly because we know that there's that psychosis tie up with cannabis.
that's right. And remember, we said there's 1,200 genes linked with autism. Or in this case, they've got 1,000. When this was published, it was a bit before that. And so these are the p-values and the odds ratios and the things of
Yeah.
the linkage with cannabis. So THC-related genes, autism, intellectual disability, and schizophrenia. So this is the overlap, the genes in the data set, differentially regulated genes in the odd ratio. So there are about twice as many genes from cannabis in the autism data set. So this is obviously cannabis, damaging the genes at the epigenetic level. So in other words, you've got the autism and some mechanisms which cause the epidemic. And this is why it's growing exponentially in the US. And this is why it's worse in the cannabis legal states. This is why it's growing in the same time space pattern as the cannabis exposure. And this is why the E values and all the causal measures are so high, because the mechanisms through the synapses, the morphogens, and the software programming of the genomes on the epigenome is controlling, disrupting this pattern. And you can see it with major brain malformations like spina bifida and severe microcephaly and anencephaly, which is no brain at all. And of course, the autism up the milder end of the spectrum. It's really obvious as you understand all this. It's obvious how this stuff hangs together. So it's not even a question. And the point is, this is well established in the science. This is well established, as I just showed you. There's lots of science about this. And I haven't even hardly scratched the surface with the depth of the scientific evidence. But what is missing is this translation from the scientific community into the general community and the general debate, especially in the US right now, with all this discussion of what's causing the autism epidemic. And as you think about the details, you can see, I think it's really silly to vaccinate pregnant women, because we know that it's been known for ages, stimulated immune system will damage brain development in the growing child. That's dumb. And the point of the vaccines is to stimulate the immune system. And we know you don't do that in pregnancy. We want women to have very quiet immune system so the brains are fine. This stuff, so I agree that that's wrong, but the evidence shows that this is cannabis driving it, especially in the legal states, and especially in those alternate communities that use a lot of cannabis. Remember what I said at the beginning? The reason we started to look at this, I've got many families with 100% hit rate, all their children are autistic, ADHD, mentally impaired, severely behaviorally disturbed, can't go to school, can't go to special school, get kicked out at the ages of 4 and 5 from school for being uncontrollable, right? And that's why we started. And that's 100%. This is not a mystery. It's only a mystery in the popular culture where they don't understand the science and the clinical reality that we see. I've got colleagues who work in ERs in Colorado. They see exactly in Colorado what I see in these cannabis using families. This is not a mystery, but it's not penetrating the popular culture. And that's what the challenge is.
Yeah, and we really have to get the media to pick this up and let the population know what cannabis is doing. So thanks, Stuart, for your time for this presentation. And we will be back with a few more after this. So you can be sure of that. So thanks for the time.